All right, and I will go ahead then and introduce our speaker for today. So I'm very happy to introduce Rachael Bay, who is at the University of California at Davis. She has lived all over the place we're talking about, including done her training in a variety of places. So her undergrad degree was at the University of Miami. Then she got a master's from Dalhousie University in Nova Scotia. And then her PhD is from Stanford, where she worked at the Hopkins Marine Station. And then she did a postdoc at UCLA. And since 2018, she's been an assistant professor at Davis, where her group studies interactions between human-induced changes in the environment and evolutionary processes. And I really like the way her work links across evolutionary ecology and climate change. And I think it uses a really neat combination of organismal biology, fieldwork, and genomic techniques to address these really timely questions. One of the honors she's received is a 2019 Sloan Research Fellowship for her work on thermal tolerance in corals, which I think is probably the basis for what she's going to tell us about today. So with that, I will turn it over to you, Rachael.
Thanks so much for the introduction, Megan, and for the invitation. I'm super excited to be here virtually anyway, to chat with you today about some of the work that my lab is doing. The first transition is always the hard one. Okay. So I've always been interested in this fact that humans can direct evolution. And we do this in intentional ways, like with crops and livestock, but we also do this in unintentional ways. You have a few examples here that I think are really neat. One of the ones I remember learning about in intro bio is this industrial melanism in peppered moths. That's now like a seminal example. But we also have more recent examples. For example, the anoles here develop different morphologies in their limbs and toe pads in order to stick to impervious surfaces that are more common in an urban environment. In urban environments. The blue tits that you see on the top right here have evolved longer bill lengths in order to access bird feeders. And the cliff swallows here have evolved shorter wings in order to veer away from oncoming traffic. So the reason that humans induce evolution is because we apply novel selection pressures that are potentially unseen in the evolutionary history of these organisms, and we intensify selection pressures that already exist. And perhaps one of the most global examples of that is global climate change. So across the entire planet, we're changing temperature regimes, precipitation patterns in the ocean, pH patterns. So I think it's pretty likely that evolutionary trajectories are bound to change as well. And one of the ways in which we study evolutionary response is to look at how organisms vary across spatial environmental gradients. So we know that environments vary over space. This is an example from California where you can see mean maximum temperature across the state. And you see some broad scale patterns, like the fact that it's generally hotter in Southern California than it is in Northern California. But you also see some smaller spatial scale patterns that create this mosaic across the landscape. And we often think that organisms should be adapted to their local climate conditions. So for example, maybe an oak tree in Southern California is more able to deal with heat waves than one that lives in Northern California. However, we also know that environments vary over time on different scales. So on daily scales, on seasonal scales, and over long periods of time. This is that same metric that's projected about 50 years into the future. And you can see that overall the landscape is getting hotter. Those red blobs are getting bigger. So when we think about how we might connect these two, this sort of spatial scales and temporal scales, the approach that my lab normally takes, and many people that study climate change take, is to try to learn something from spatial environmental gradients, try to understand underlying patterns and mechanisms of environmental adaptation, and then use that to predict something about the future. So maybe that's future distributions of species, the adaptive capacity under climate change regimes, or vulnerability to climate change. But it's also not quite that simple because the degree of local adaptation can also impact a species level response to climate change. So we have sort of a toy species here, a toy example here, where you have a species that inhabits a broad environmental gradient. You can imagine maybe on the x-axis temperature, but there is an optimum. So in this case, this species is most fit at intermediate temperatures. So that might be where you have the highest abundance, or if you have experimental tests where the species has the highest fitness. And in this make-believe species, every individual has the same tolerance. So the same breadth of tolerance, the same optimum. If we lose some fraction of that species niche because of climate change, so maybe the low end of that temperature gradient no longer exists, we still have quite a bit of that niche left. So the species can still exist in the rest of the niche. However, if instead of one sort of homogeneous species where every individual has the same fitness in the same environments, we have a series of serially adapted populations where each population has a different climate optimum that make up the overall species curve. Climate change response might look a little bit different. Now, when we lose part of that climate range, we lose maybe a whole population, maybe particular genetic variation. And so we have a sort of different outcome when we think about both adaptive capacity and genetic variation overall, when we think of climate change response from an evolutionary perspective. My lab works in a lot of different systems. Primarily, we're interested in climate adaptation in marine systems. And I think that marine systems are particularly interesting because I think there's reason to believe that adaptation might work a little bit differently in many marine systems than it does in terrestrial systems. And the primary reason for that is because water, rather than the air, is the medium in which all of the organisms live. And this has a couple different consequences. One is that many of the organisms have this sort of small propagule life history stage where they exist passively, or at least mostly passively, in the water column. And this allows them to disperse quite far, which leads to high gene flow potential. In general, we see shallower environmental gradients, and we see more complete niche filling, again, because of this dispersal potential and faster range expansions. On the other hand, many marine organisms also have a biphasic life cycle where, although they have this propagule stage where they can travel very far, most of their life is made up in this sessile stage. So you see the examples of coral and seagrass here, where they have to sort of deal with any environmental conditions that they come across without moving. So you have these like really different needs of the different life history stages. And so today I'll actually talk to you about two of those systems. They're the ones that you see here. Some coral work, which is mostly what I did in my PhD, and then some more recent work that we're doing in the lab now on seagrass. So we'll start with corals, and I think corals are a really important system to look at climate adaptation in because they have really, over the past decade, become the poster child for climate maladaptation. Corals are these large colonies made up of millions of clonal animals called polyps. And within the tissue of the polyps, they have these algal symbionts that are photosynthetic and provide the coral with nutrients. However, this is a sort of tenuous partnership. So when stress gets high, the symbionts get expelled, and functionally the coral starves to death. And that process is known as coral bleaching. So once the symbionts are expelled, there you no longer see the color in the coral's tissues, and you see this sort of stark white phenotype. So we've seen a lot of photos like this in the news over the last 5 years, especially with recent El Niños. And we know that warm water events can lead to this sort of massive bleaching. But when I see a photo like this, I do see this sort of large scale massive bleaching, but I also see a lot of phenotypic variation here. So you have a lot of corals with that stark white phenotype, but right next to them, you have individuals that actually look pretty healthy. So one of the main questions that drives our research here is, what is the difference between those individuals? Why do some corals seem to bleach completely while their neighbor survives the heat wave just fine. And we know that thermal tolerance or coral bleaching susceptibility varies both among species and among individuals within a species. These are two photos I took from in the field during a bleaching event a few years back. On the left, you see two different species with really different bleaching phenotypes. As you can see, the one on the sort of most left is just completely white, completely bleached, while you have that pink colony that might look a little pale, but is mostly doing pretty okay. On the right, you have two individuals of the very same species planted almost touching each other and have experienced the same heat wave in really different ways. So the colony on top still has full pigment, seems to be doing fine. I can even see some of its tentacles sticking out because it's trying to feed. And on the bottom, you have a completely bleached colony. So I think this just really presents an interesting puzzle. Why do you have that phenotypic variation? What is the basis for that? And a lot of folks have been looking at this. Of course, it has become an incredibly urgent question over the past years. And the complexity of it is that there's a lot of different answers. So one answer is plasticity. We know that thermal history of an individual can affect its response to future acute heat stress. We know that those algal symbionts that live within the cell of corals are incredibly genetically diverse. So that's actually an entire family of algae. And we know that the different species within that family have different characteristics, provide their hosts with different nutrient levels. So the symbiont genetics can have a big impact on bleaching response as well. There's increasing information showing that the microbial community can have an impact on bleaching response, and the sort of stability of that community really matters. And the one that my lab is most interested in is thinking about the genetics of the coral host. And so genetic variation in the coral host might lead to different bleaching outcomes. The study system we'll mostly talk about for coral work today is this really nice, what I like to think of as a natural laboratory on Oku Island in American Samoa. And the reason this is such a nice place to look at thermal tolerance is that the reef structure here creates these back reef lagoons that have really different temperature profiles. So we'll talk about throughout this talk the highly variable pool and the moderately variable pool. I think their names are kind of intuitive, but the highly variable pool has these like large magnitude swings in temperature. You can see the temperature profile for about half a year on the bottom, where the temperature swings from a little lower than 28 degrees to maybe 34 degrees. We've even recorded 35 or 36 degrees if it's a really hot day and a low tide. On the other hand, the moderately variable pool also has these swings in temperature, but they're dampened. So it rarely exceeds 32 degrees. The dotted line you see here at about 30.5 is the nominal bleaching temperature for corals in this region. So both of the pools actually exceed the bleaching temperature regularly, but the highly variable pool does this by 3, 4, or maybe even 5 degrees sometimes. So that's pretty dramatic. And even in times where we've seen sort of short, small magnitude changes where you exceed that bleaching temperature, we often see that the reef remains healthy. So until the past few years, we hadn't seen many major bleaching events in that pool at all. So the question is, before that, how did this reef remain healthy? And we also know there's a lot of temperature variation within each of those pools. So each of the points here is a different coral colony that we put a temperature logger on that could measure temperature every 10 minutes. And we're plotting the amount of time each individual spends above 31 degrees. This is close to that nominal bleaching threshold. And you can see the difference between pools really drastically, but you also see quite a bit of variation within each pool, and especially within the highly variable pool. So first we wanted to just establish whether we saw adaptation between the two pools. And we did this with a reciprocal transplant. So the nice thing about corals is that because they're clonal, you can fragment them and do multiple tests on the same genotype. So we're able to fragment colonies from the highly variable and moderately variable pools and make these replicate transplant grids where each grid had a representative from each genotype, and we were able to distribute those grids across the highly variable pool and moderately variable pools. So the first question we wanted to ask is just what happens to thermal tolerance itself. And we did this experimentally with these portable heat stress chambers where we functionally just heat corals up in a beer cooler and see what happens. In the histogram I'm showing you now, I'm showing you thermal tolerance on the x-axis. And the important bit is that higher numbers mean higher thermal tolerance. And so this histogram shows corals that are from the moderately variable pool transplanted back into the moderately variable pool. And this is just showing you that distribution. On the other hand, if you look at corals that are from the highly variable pool transplanted back into the highly variable pool, you see they have higher thermal tolerance. But this is a mix of fixed and plastic effects. So what we really want to know is what happens when we take individuals from the moderately variable pool and transplant them into the highly variable pool. And we left them there for about a year. And what we see is an increase in thermal tolerance, but it doesn't quite match that of highly variable pool natives. So in summary, what we see are both fixed and plastic effects that are contributing to that thermal tolerance difference between pool. And those fixed effects could include things like adaptation, among other processes. We looked at a couple different other traits, and we see some similar results. Here I'm showing you survival for individuals from the highly variable pool on that red line, individuals from the moderately variable pool on the blue line. And it doesn't matter where we transplant them into. So on the x-axis is their current growing environment. The individuals from the highly variable pool grow better no matter where we put them. So I think like an important question to ask yourself at this point is, okay, these individuals from the highly variable pool survive more and they're more thermally tolerant. Why would we expect variation in the system at all? Shouldn't these high surviving thermally tolerant corals just take over the world? And the answer to many things like this in evolution is trade-offs. And so when we look at growth, we think we see at least some weak evidence of a trade-off. So individuals that do survive really well in that highly variable pool tend to be slower growers. And for corals, that slow growth could lead to a number of outcomes, including maybe they're less good at competing for light and resources. Maybe they have lower reproduction because functionally they have fewer polyps to reproduce. So you can imagine how that would be a fitness trade-off for these corals. But these pools are really close together, so less than a kilometer apart. Gene flow is functionally infinite between the two pools. There's no real population level differences. So how do we maintain these allele frequency differences? And this is the model that we sort of assume. Corals are broadcast spawners, so they release their gametes into the water column where they meet up and they stay in the larval phase for potentially weeks. And so you have this mixed larval pool of larvae from both the highly variable and moderately variable pool, and probably a mix of the two. And then you have post-settlement selection that leads to these trait level differences in the two pools. So after the larvae have settled, maybe a heat wave comes through. We know that there's really high mortality at the juvenile stage. So you can imagine small juveniles without maybe that are less well adapted to hot environments might lose out at that stage. But we wanted to look at the allele level as well to see if we saw any genetic differences between these corals. And so we sequenced individuals. At this time, we were using RNA-seq because genomic sequencing was not yet available in corals, and that's how much I'm dating my dissertation right now. But we sequenced individuals across the two pools, and we were able to look at 15,000 SNPs encoding regions of the genome. And we used two different types of analyses to look for candidates of temperature selection. The first is a pool-based analysis, so that's FST outliers. Here we're looking for loci that are more differentiated than we would expect based on sort of the genome-wide average between the two pools. But we also have individual temperature variation and individual genotypes, so we can connect the two and do a sort of G by E correlation. And using those two analyses, we were able to find about 114 SNP candidates for selection that were significant in both analyses. And when we look at how alleles are distributed across the highly variable and moderately variable pools, what you see is that the rare alleles become common in the highly variable pool. So the model that I think about for this is that this area, the highly variable pool, is geographically small but highly selected. So alleles that are rare in the population on a whole become more common in that location. So what we've learned is that thermal tolerance is really polygenic. We know this in a number of systems now. It's a product of small effects across many genes. We also saw that there are trade-offs which might suggest an optimal number of HV-like, or what I'll call thermal tolerance alleles from now on, in different environments. So if we make sort of a verbal model of what we think is going on here, we might expect something like this, where you have a number of thermal tolerance alleles, and the number that you need is dependent on your current temperature. And so when you have the optimal number, you have the highest fitness. The further you stray from that optimal number, the lower your fitness. So now that we have that verbal model, we wanted to see if we could turn that into some sort of prediction. And in order to do that, we looked at corals in Rarotonga, which is in the Cook Islands. It's about 21 degrees south latitude, so it's quite a bit cooler than Ofu. About 2 degrees cooler in terms of bleaching temperature, and near the southern limit of like open ocean coral growth. So it's like a cool water coral reef. And so we sampled some additional Acropora hyacinthus individuals from Rarotonga, and we genotyped them at the same SNPs that we had in the Ofu system. If we look at the first principal component across all of the SNPs, we see what is basically an isolation by distance signal, so separating the two islands. Rarotonga looks different than the highly variable and moderately variable pools, both on Ofu. However, when we look at just our candidate SNPs, what we see is that that highly variable pool looks different than moderately variable pool and Rarotonga together. So this gives us a little bit more confidence that those SNPs are doing something special in the highly variable pool. And so next we use population genetic simulations to try to tie the frequency of candidate SNPs to some sort of demographic process. So we start with allele frequencies and a rough estimate of population size. And this is, for those of you population geneticists, this is basically like a Wright-Fisher-like simulation, except that we need absolute fitness rather than relative fitness. So the way that we accomplish that is to have probabilistic survival. So you'll remember that curve I showed you where like the number of thermal tolerance alleles you have dictates your fitness in a particular environment. We use a curve like that to invoke probabilistic survival based on the number of thermal tolerance alleles you have and the current temperature that you're in. Everyone has the same probability to grow and reproduce, and then we can recalculate the new allele frequencies and the new population size and start the next generation. So we need to invoke warming somehow, so we use real temperatures from Rarotonga. These are what the temperature projections look like for Rarotonga at that time, and you can see the different emissions scenarios listed here. So the two blue scenarios are our more mitigated scenarios where you see very little change in hottest temperature. And then the orange and red scenarios are more severe scenarios. In fact, the red scenario is our sort of business as usual scenario. And that's about 2.5 degrees hotter than at the end of the century than now in Rarotonga. And so when we use this to determine the sort of fitness functions, this is what we get for fluctuations in population size over time. So you can see that the mitigated scenarios are able to maintain coral populations by the end of the century, while the more severe scenarios see extinction, and the most severe scenario sees extinction quite quickly. And there's a lot of ways that you can vary parameters here, but I think the most important bit here is that the rate of warming compared to the potential rate of changes in allele frequencies can really make a difference. And even in the scenarios where we see persistence to the end of the century, we can ask ourselves, is adaptation happening or are we just not exceeding the environmental niche for the species? And what we see when we look at the shift in allele frequency of those thermal tolerance alleles is that we do see adaptation happening, especially in that 4.5 RCP, where we see this increase in the frequency of thermal tolerance alleles. So that's maybe some like vague suggestion that evolutionary rescue might be happening. So in our simulations, adaptation's rapid enough for moderate, but not severe ocean warming. So now that we have this simulation framework built, we can ask more applied questions. In corals, one of the ones people are really interested in is, what about assisted migration? Can we take corals from warm adapted populations, place them in cooler populations, allow their thermal tolerance alleles to spread within those populations and rescue them from warming? So really quickly, I just want you to pay attention to RCP 6.0 because that will just be focusing on that one. And we're going to simulate assisted migration where we add individuals that look like HB OFU individuals to the population every generation. So we add between 0 and 50 migrants a year. At 0 migrants a year, the curve is exactly the same as you saw in the last slide. You see extinction. But at any level of assisted migration, you see persistence, and the level of the population size at the end of the century depends on how many corals per year you're adding. I think at the surface of this seems like a really optimistic message. Assisted migration seems to be working, at least in our simulations. However, I think the magnitude is important for context here. So what we're talking about is seeding 1% of the population size of adult reproductive corals each year until the end of the century. There's a lot of logistical challenges, not only human effort, but the source of those heat-tolerant colonies. So while I think this does give us some like tools and intuition to think about assisted migration, I think that we have to be careful in thinking about this as a super optimistic message. But there are a lot of ecosystems that are being restored right now, and I think a lot of places where genomic data can inform those restoration efforts. And so we're going to switch gears and talk about one of those. This is eelgrass, or Zostera marina. It's a really common foundation species pretty much globally, and it's a really commonly restored species. And one of the interests in restoring is to do that so that new eelgrass beds will be more climate resilient. And functionally, when we're asking that question, we're asking how predictable is response to climate change based on genomics, or how transferable are genotype temperature relationships. So I've been doing this in collaboration with Jay Stackwitz and Rick Grossberg here at UC Davis. Lauren Scheibelhut was our postdoc that worked on some of this work, and Brooke Benson and Carolina Zabinski are the graduate students that are working with us now. So just a little bit of natural history here. Eelgrass is widespread across the Northern Hemisphere. It's important nursery habitat for fish and invertebrates. And there's also been some recent interest in its function in carbon sequestration. However, we've seen eelgrass beds declining from a range of causes, including water quality, climate, disease, and development. But eelgrass beds are actively restored, and they do this by transplanting individuals from healthy meadows. So one of the questions can we do this in a more informed way in order to increase resilience to climate change? And from the perspective of a sort of ecological genomicist, can we use genomic data to predict something about how those plants will respond to climate change? And so functionally what you're asking there again is evolutionary predictability, and we can think about that as across parallel environmental gradients. So our first thought was to look across latitude. Because eelgrass is so widespread, we actually have replicate latitudinal gradients here. So the easiest thing to do might be to say, okay, well, do southern populations across all of the coasts harbor the same genetic variation? And if so, maybe that's a hint that that genetic variation is better suited to deal with hotter temperatures. This is the only slide I'll show on this data. Happy to talk about it more. But basically what we found is that 93% of the SNP level variation is fixed within one ocean basin or the other. And in fact, there's very little shared genetic variation across any of these populations, making SNP level parallelism really hard to find in this sort of data set. But if we're talking about sort of feasibility of restoration projects, we're also not planning to do sort of basin level transplants. So maybe it's a better idea to look more locally anyway. So what we did was to zoom in on a local environmental gradient here near the UC Davis Marine Lab in Bodega Bay. So we have two adjacent environmental gradients here, or temperature gradients, one in Bodega Bay and one in Tomales Bay. In both cases, the mouth of the bay is cooler than the back of the bay. I can show you some temperature data here. That's Bodega, so you can see that that green line is clearly warmer sort of throughout the time period than the other lines. And in Tomales Bay, you see a similar pattern where the sites near the back of the bay are warmer than sites near the mouth of the bay. And we know from previous work in Jay's lab that there is local adaptation across the bay. So this was a reciprocal transplant done across 3 of the sites in Tomales Bay. And when you look at shoe count, you see that the home site always has an advantage. So the native individuals always have higher shoe counts. And some follow-up experimental data showed that temperature was at least one of the important drivers of this pattern. We did a common garden experiment, and we see similar sort of population level impacts of temperature, or at least differential responses to temperature. Heated plants always had higher growth, at least when we're looking at above ground growth. But individuals from this Blake's population, which you see in purple, had a larger response to heat stress, at least for growth, than individuals from the Millerton population. So there's differential responses here. And if you look closely, you can see a sort of shift in rank order of the 2 different populations, and that's also apparent at the individual level. So we can compare the performance of the same genotype across multiple years in the same condition to see how predictable the phenotypes are. So when we look at genotypes grown at ambient temperature from across year 1 and year 2, we see a positive correlation regardless of the population they're from. However, when we look at genotypes grown in ambient versus elevated temperature, there's no correlation whatsoever. So there's this shifting in rank order of genotypes when grown in ambient versus elevated temperature. When we think about this, what this might mean in terms of predictability and genetic architecture, potentially there's a different genetic mechanism for growth at elevated temperatures. But certainly if we measure, if we establish our genotype environment and sort of model at ambient temperature, we won't be able to predict performance at elevated temperature. So that makes it a little bit challenging. So what we want to do is see if we have shared signals of selection across the 2 different bays. Again, you have temperature gradients across both bays, and we use a program called LFMM, which looks for correlations between genotype and environment as SNPs across the genome. And when we're looking in just Tomales, this is what we see. So we see some peaks where SNPs are significantly correlated with temperature. They're kind of scattered across the genome. And when we look at Bodega, we see something similar where there are some significant SNPs that are kind of scattered across the genome, but none of those SNPs overlap between the 2 different analyses, not a single SNP. However, there is overlap at the gene level. So there's a possibility that we have sort of multiple mutations that lead to the same function in some important genes. But so this is not a good signal to suggest predictability of any kind, right? If we have no overlapping signals of selection, I wouldn't necessarily expect us to be able to predict across phase. But we wanted to explicitly test that hypothesis anyway, so we just went for it. And we used a sort of polygenic framework using a random forest model. And what we did is that we predicted temperature from the genomic data using the random forest model, and we compared that to observed temperature. Now, what the random forest model needs is a set of candidate SNPs. So these we can get from our LFMM genome scans. We also need to train the model, and then we predict on another data set. So there's a lot of different permutations we can do of that. First, let's take the candidate SNPs that we found in the Tamalis data set, or in the Tamalis, across the Tamalis gradient. And when we take candidate SNPs from Tamalis and we train on Tamalis and we predict on Tamalis, we do a really good job. So as long as we're staying within that population, we do a great job. But if we use the candidate SNPs from Tamalis and predict on Bodega, we do a pretty bad job. So this is in line with what you saw with the lack of concordance with the genome scans. We have low predictability in Bodega. And this is similar to some of the stuff you've probably seen in human populations where polygenic scores are not necessarily transferable across populations for a number of different reasons. However, again, you can make any sort of any number of combinations between the candidate SNPs, the training set, and the prediction set. So we did those just to make sure that we knew we had no predictive power ever. And so here I'm showing you just the same thing I showed you on the last slide. When we take LFMM Tamalis SNPs, we use those to train in Tamalis. So these are the bottom two rows here, and then predict in either Bodega, the second to bottom row, or Tamalis, which is the bottom row. So you see that non-significance in the Bodega population in that middle row there. However, if we take SNPs that we discovered in Bodega, we see a pretty different pattern. We see that SNPs discovered in Bodega are able to predict temperature regardless of where we're using them. So this is not like what we expected. We didn't expect to be able to take those SNPs discovered in Bodega and predict Tamalis temperature, but we actually do a pretty good job of predicting Tamalis temperature with those Bodega SNPs. So it's possible in that Bodega SNP set, there's something that is helping us predict temperature, potentially amongst a bunch of covariating noise. So we're going to switch gears a little bit and talk about one of those potential signals, and one that seems to be especially sort of standing out from the rest. If you were looking closely at my Manhattan plots, you might have noticed this like pretty weird line that showed up where you have a bunch of linked SNPs on chromosome 1 that are significantly associated with temperature. That didn't show up in Tamalis, but also if you were paying really close attention, you might have noticed that our temperature gradient in Tamalis is much broader than our temperature gradient in Bodega. But we can truncate our temperature gradient in Tamalis in order to perfectly overlap with that in Bodega, so just by using the first bit of it. And when we do that, that same links set of SNPs shows up, although just barely over the significance threshold that we're using. So let's zoom in on just that section of the chromosome. What we're going to do is make a PCA of chromosome 1 and a PCA of just that target region. So when we look at chromosome 1, we see what we might expect genome-wide. Bodega and Tamalis are distinct. This is an isolation by distance signal. But when we look at that target region, we see 3 really distinct blocks. And this is a sort of hallmark of a supergene or potentially an inversion where you see no recombination between those SNPs. So these functionally are 3 genotypes. The inversion is 5 megabases long, but there's very little recombination within it, if any. So we can look at genotypes of that inversion across Bodega and Tamalis. And again, our sample sizes per site are pretty small. And so we can't see any dramatic decline within Tamales or Bodega, but what we do see when we test against Hardy-Weinberg is that there are a lot more heterozygotes than we would expect based on Hardy-Weinberg. So while we don't have a clean sort of cool to warm cline in this inversion, maybe there is some selection going on where you have this increased number of heterozygotes. So we can go back to our phenotypes to try to understand the fitness effects of this inversion. So now I'm showing you inversion genotype on the x-axis in the two different populations, and this is information from our common garden experiment. And on the y-axis, shoot biomass. And what you see is that individuals from flakes always grow more, but heterozygous individuals also always grow more. So within each population, heterozygous individuals have the highest shoot biomass. So this is potentially a fitness advantage, not necessarily directly, because there are a lot of other growth metrics that contribute to fitness. But not all sort of phenotypic correlates are as clear. So here's another one. One thing that happens to eelgrass is that it has this wasting disease that's caused by a protist that actually decimated Atlantic eelgrass populations in since the 30s. We expect sort of like a 90% decrease since in the last century. And the lesions look like the picture that you see here. And we see a really different signal here. We see an interaction between population and genotype. So actually in the Blake's population, there's no difference between the three genotypes, but in the Millerton population, the heterozygotes have the lowest severity of these disease lesions. So there's potentially an interaction with like some genetic background or something. But it does seem that the genotype of these inversions affects performance, where in both of these cases, it seems like there's a good case to be made that heterozygotes might be more fit in these environments. So just to wrap up, we see everywhere really that thermal tolerance varies among individuals and populations. In the coral simulations, we showed that the rate of warming really matters for population persistence. In the seagrass, we show non-parallel adaptation, and I think that's really going to make transferring these genomic scores really difficult. I do think that supergenes are an interesting way to look at adaptation and potentially more predictive because you have the ability to put together these co-adapted gene complexes, but in our system, they seem to have really complex effects on performance. And I'll just wrap up by thanking the co-authors on all of this work, as well as funding mechanisms, you for coming to listen to me, and I'd be happy to take any questions.
Definitely the weirdest part of Zoom seminars is the part right after the talk.
Is anyone still awake?
Oh, definitely. A question already came in the chat, and I guess I'll just say folks are welcome to either raise their hands or unmute or put a question in the chat, and I can relay from the chat. So question starts with, thanks for a great talk. I agree, it was a great talk. And then it has some questions about coral reefs. Wanting to know about the nearshore land cover on Ofu and Rarotonga, like what are the dominant tree species, and has land cover changed, and is that related to bleaching?
Yeah, great questions. I know very little about trees. Both islands seem to me like they have a lot of palm trees and maybe some like big type things, but like I don't know anything about trees, so sorry. And both are volcanic islands, so you have a sort of small strip of flat land and then very steep rainforest. And so I don't think land cover is high. There's very little agriculture, at least on Ofu. There's more agriculture on Rarotonga. I don't think much has changed since coral bleaching has started, mostly because the vast majority of coral bleaching started in like at least the severe global bleaching events started in like 2015. So we're talking really recent history. And I don't think there's been a lot of change in agricultural practices on either of those islands. And most of the usable agricultural land would have been used long before that because it's so little.
Gideon.
Hi, Rachel. That was really fun and really interesting. I was really curious about that inversion. It's interesting that you don't see it if you look across all of tamales, but you do if you just look at the same color gradient. Have you looked in the genotype data to see what the frequency of the inversion is farther south? Like, is it there, or but just like at frequencies that don't correlate with temperature, or is it not there at all?
Yeah, so take a look at this map. Here I'm showing you the 3 different inversion genotypes, right? So even at the back of tamales, you see all 3 genotypes. But so I think part of the correlation is just like our sample size per site is not good. And so you see this site by the mouth kind of driving that pattern in the cooler populations. But then this site, which is Millerton, has more of that AA genotype. So there's not a clear line across the bay, but it is everywhere. We've been doing it's super confusing. So we developed like a restriction genotyping assay for it. And so we sent some undergrads up and down the California coast last summer to genotype other populations. And we find no polymorphism anywhere else for this inversion. San Diego is fixed for that BB genotype. Every other population we've tested is fixed for the AA genotype. And Bodega and Tamales are the only place that is polymorphic. And I don't know what the heck is going on.
And wait, so just so A is the cold phenotype, or the cold allele?
Yeah.
Okay.
I mean, if there's a cold allele.
Right. Yeah. Wow, that's super cool. Thanks.
Yeah, it's weird.
I taught about heterozygote advantage yesterday, and so was starting to try to write exam questions in my head during your talk.
And this is maybe the first time I've like directly tested Hardy-Weinberg like in my own research.
Yeah, Alexander.
Hello. First, I want to say thank you for the talk. Very interesting. I'm a big coral nerd, so I feel bad. I came a bit late and I missed most of the coral stuff. But at the tail end of it, I heard you talking about potential like shifts in populations for corals. And I was wondering if you've done any research or heard anything about like the Arabian corals, because I found that like a lot of Arabian corals have much higher thermal tolerances than ones that we're finding in the Indo-Pacific and Caribbean. And there's been some ideas about potentially using those to like stock populations of corals outside of the Arabian Gulf, and potentially helping like transplant those with like native corals in new ranges. And I was wondering if you've heard anything about that or have any info you might like to share.
Yeah, so we don't do any work in Arabian corals, but I know a lot of folks who do. And I think the cool thing about that is that like they reach the highest temperatures of any coral population. So that's the sort of logic behind using them, or at least understanding them, to understand the basis of thermal tolerance. There is some talk about like, should we be transplanting those corals outside? I am generally pretty hesitant about like long distance transplantation, because I do think that like when we focus on a single trait like thermal tolerance, we're missing a lot. By definition, like that's how evolution works. There are trade-offs. And so I'm of two minds there. Like on one hand, there are evolutionary trade-offs if we focus on breeding or assisting migration for a single trait we might be doing a disservice to other traits that are important in local environments. On the other hand, it is the single trait that is like really determining coral survival right now. So there's a little bit, I have mixed feelings about that, but there's a lot of great work trying to understand the mechanism just of survival in the Arabian Gulf.
Have you also, now tying back towards your seagrass study, I saw you were looking at single nucleotide polymorphisms, but have you done any like proteomic data to discover if there's any like proteins specifically associated with differential tolerances?
Yeah, we haven't at all. We haven't done any stuff, but there's been some gene expression studies looking at differential transcription, but I don't even know of any proteomic studies.
Yes, ma'am.
Sounds hard.
Okay. Yeah, it's tough. I was working with a team over the summer trying to do a proteomic study with coral, and we ran into a lot of roadblocks. It was very difficult.
Yeah.
Yeah. All right. Well, thank you very much.
Something that just made me, or like those questions were making me wonder about is with the eelgrass, the thermal tolerance, right, with this heterozygote advantage, that there was maybe like was it a trade-off between the thermal tolerance and disease resistance?
So there's it acts different based on like population background, right? So I'm actually not separating out temperature treatment here because it looked there's no difference between the temperature treatments. But the warmer population, which is Millerton here, has that genotypic difference. So I don't know if that has something to do with like interaction with other genes in the genome. There is a decent bit of structure across Tamalis. So there's like plenty of, not fixed, but like allele frequency differences between the populations.
Yeah, and part of why I was thinking about that is like going back to what you said, like trade-offs are everywhere. And I share your hesitancy about moving things a long way without really knowing what else might be changing along with thermal tolerance.
Yeah. Yeah, so I'm hoping that we can dig a little bit more into this. We've got a whole suite of traits that this is our sort of first look at the connection between the inversion and the trait data. We've got a whole suite of traits that hopefully we can make a more complete picture of fitness effects with.
I have a question. I've been sitting here. Thank you, Rachel. That was really interesting. I have many questions, but I guess just informed by your response to Meg and thinking about population structure as a confounding factor in Tamalis Bay. Before you talked about the heterozygotes, the heterozygote patterns here. That's what I was thinking about, and sort of how you would build a predictive model of this sort of adaptation when you also have population structure driving these allele frequency shifts. And I know that this is more like a broad question, but I wonder what you think about that kind of practical problem of accounting for all of these factors.
Yeah, so I actually think the population structure is one of the reasons that the bodega SNPs might be more predictive than the Tamalis SNPs. I can actually show you, let's see if I have a plot of population structure here. So, I mean, isolation by distance along Tamalis is super strong. And it co-varies with our temperature gradient, right? So a lot of what we're picking up in a scan that looks at temperature is probably just population structure. And because the bays are oriented opposite from each other in terms of population structure, it makes sense that then those would not predict bodega temperatures. But population structure in bodega is pretty limited. So I wonder if you're doing a better job picking up a signal of temperature in bodega, and that's why those SNPs are more predictive, even though they don't overlap with Tamalis. So that was a sort of long way around, but I think that what I think is that there still might be important SNPs, but we have to look across like multiple environmental gradients and find the ones that find general rules that we could actually use for any sort of application, if they exist. Who knows?
Thank you. And then I see this parallelism challenge when you don't have the shared variance. So yeah.
That was
Very
disappointing.
Thank you, Rachel.
Yeah.